Welcome students, welcome to Priya's Life Science Academy. In the previous class, we have understood about Mendel's three very important laws, that is law of dominance, law of segregation and law of independent assortment. In today's class, we will read some more important topics and terminologies, okay? So pay attention and listen carefully. If you have any doubt about the previous classes, you can always comment below and I'll be happy to give you a suitable answer. So let's start. So from the previous experiment, what Mendel found? Mendel found that the inherited traits were either dominant or recessive, okay? Dominant allele expresses over the recessive always, because we have seen that capital T, if a plant is of genotype capital T small t, then the plant will be tall, because dominant alleles always expresses over the recessive, okay? And one allele is dominant over the other, capable of masking the recessive allele. We know that, and recessive allele can only be expressed when it is present in double dose, that is in homozygous state. We have already encountered these things. So this is the thing. So this is the purple color flower, that is capital P, capital P. This is the white colored flower, small p, small p. This is also the purple colored flower, capital P, capital P. These things we already know that when the dominant allele is in double dose, it's going to express itself. When the dominant allele is in single dose, it will mask the recessive allele and it will express itself. It is purple in both cases. This is homozygous dominant. Why it is known as homozygous, we already know. Two similar kind of allele. This is heterozygous dominant. I want you to learn every terminology very clearly. That is heterozygous dominant, okay, because hetero means two different allele, that is capital P, one is small p, and this is homozygous recessive, and homozygous means two similar alleles. So these things we already know. Homozygous recessive. Now we have to see, is there any other kinds of phenotypes other than dominance and recessiveness? Not all traits are purely dominant or purely recessive, okay? In some cases, neither they are dominant, neither they are recessive. Now that is known as incomplete dominance, okay? There is a establishment of a intermediate character. Intermediate character, a character that is neither dominant nor recessive, in between dominant and recessiveness. Now please look into it. Now what is incomplete dominance? Incomplete dominance means the pattern of a gene expression in which the phenotype of a heterozygous individual is intermediate between those of the parents, okay? That is, the one allele is not completely dominant over the other. For example, you know, if genotype, if the genotype of a plant is capital P, small p, what will be the phenotype? You will say that it is purple, but no, it is not always purple. It can be an intermediate color between them. It can be pink, okay? So let's look into the example. So a third new phenotype appears in the heterozygous condition. Not always the dominant allele overpowers the recessive allele. There is another third intermediate character that will appear. For example, you can see here capital R is red, small r is white. That's very normal. And what is it? Capital R and small r. Capital R is dominant allele, the small r is recessive allele. From your previous perception, you would think that because it's capital R is there and it will dominate over small r and it will be red. But no, it's not red. There will be an intermediate color between red and white, that is pink. So in this case, the heterozygous condition, there will be an intermediate character, there will be a new phenotype that is pink. This is known as incomplete dominance. The dominance is not the capital R is not properly dominant over small r. That is why it is known as incomplete dominance. There is a color in between red and white. This example we can see in many flowers: snapdragon flowers, roses, carnations. The most common example are roses. We can see multiple color of roses because of this incomplete dominance. Okay. Now, why does it happen? Now the question arises. We have already read that capital R is the dominant allele. It can produce the red color, and when it is combined with the small r, the capital R is dominant. It will mask the expression of the small r. But why is this not the case in here? What happens, that individuals with a single r and a small r, okay, this individual, the single capital R is not able to make enough red pigment to produce the red flowers. That's because there is a single dose of R with the small r. This capital R is not capable of producing enough red pigment to give the flower a red color. Okay, so that is the case. So we will look into the cross. Suppose this is a red flower, capital R, and this is the white flower, R dash, or you can also write it as small r, smaller. If you cross it, I told you how to do the Punnett square. All the flowers are capital R, small r, and have you looked their phenotype? They are all pink. They are not red, as because there is a capital R present and small r. You will think that because of the capital R the flowers will be red, but no, the flowers are not red. The flowers are pink in color because it's a case of incomplete dominance. Okay, and the individuals that are white produce no red pigment. Yes, the individuals who have the smaller or r dash, they will be white in color. So this is also a case of genetics. Not all the phenotypes are completely dominant or completely recessive. There is a phenomenon known as incomplete dominance, where the capital R is not totally dominant over the small r. Okay, now this we have seen in F1 generation. Now let's see at F2 generation. So when we self-cross the F2 generation's flowers, that is capital R, small r, or capital R dash, what we can see? We can see that there is one flower with capital R, which is red. There is one flower, there are two flowers, capital R, small r, which are pink, and there is one flower, smaller, that is white. Same type of cross which we have looked in monohybrid cross. So you can see from here very clearly that whenever small r comes together, the flowers are white. Whenever capital R comes together, the flowers are red. And whenever there is one single capital R and small r, the flowers are pink. Okay, to get a red flower, there must be presence of two capital R. Here the capital R is not able to make the full red pigment. So the capital R is not totally dominant over small r. That is why it is known as incomplete dominance. Okay. So the phenotypic ratio changes here. First we have seen in monohybrid cross the phenotypic ratio was 3:1, but in this case the phenotypic ratio is 1:2:1, because one is complete red, two are pink, and one is white. Okay, so this is the case of incomplete dominance. I hope you have understood. This is the P generation. We have crossed red flower, white flower. We got the pink flower in the F1 generation. Then we again self-crossed the pink flowers, and we got one red, two pink, and one white in the F2 generation. Okay. Now I'll present you a situation. Is there a condition, or is there a possibility that two alleles of a gene can both be dominant together? Yes, there is a possibility where we can find the codominance. Okay, the condition in which both alleles in a heterozygous organism are expressed. We are getting so much concepts about heterozygous. First, we have seen that capital P, small p is purple because of the dominance. Okay, then we have seen that capital R, small r is pink, one intermediate color, that is for incomplete dominance. Now we will see one very important phenotype, that is both the alleles can be expressed, we will get mixed kind of phenotype, that is known as codominance, where both the alleles are getting chance to be expressed. In the first case, because there is a dominance, the capital P was only expressed. In the second case, you are getting a intermediate character. But in third case, there is a chance for both the alleles to be expressed fully. They both are dominant. Both are dominant. Okay, so in codominance, in heterozygous condition, where both the alleles will contribute to the phenotype. Okay, in case of heterozygous condition, both the alleles will contribute to the phenotype. It's very, very important. The two alleles don't blend, but are rather both present in the offspring. They do not blend, both are present in the offspring. Okay, examples are check-bred chickens or own horses. Here we can see the codominance. Okay, here the cross between black chicken and white chicken. Black chicken's genotype is capital B, white chicken's genotype is capital W. When both are crossed, we can get all the checkered phenotype. Okay, both the white and black colors are present in this phenotype, in this offspring. Okay, so capital B and W both are expressed in the offspring. That is why the chicken is a mixture of black and white color. There is no blending of the color. Black and white are not mixed into a gray color, but both the colors are present separately. You can see the white color in the chicken as well as the black color in the chicken. So this is known as the codominance, where both the alleles are expressed. Okay, now this is the example of roan cattle, where both the brown and white colors are there. In the variegated clover, in the heterozygous condition, both the colors are present. Okay, white and green. This is the concept of codominance, where both the alleles are getting chance to be expressed. Okay, now we will see codominance in humans. Okay, the heterozygous condition, both alleles equally expressed. We have already seen that in codominance, in heterozygous condition, both the alleles will be expressed equally, and this is the case in sickle cell anemia in humans. Okay, where the normal red blood cells are capital N, the sickle cells are SS. Okay, and in case of one genotype, it is NS, the mixture of normal red blood cell and the sickle cell red blood cell. This condition is carrier condition. Okay, and they are in heterozygous condition. Okay, here you can see the presence of both good round red blood cell and the sickled celled red blood cell. Okay, so this is the case of codominance in humans. In case of sickle cell anemia, you can see these are the patient where all the red blood cells are sickle celled. They are known as carriers, where some of the red blood cells are round and some of are sickle celled. Okay, so both the alleles are expressed. Normal alleles are also expressed, and sickle cell alleles is also expressed. So this is the case of codominance in humans. So I hope you understood what is dominant allele, what is dominance, what is incomplete dominance, and what is codominance. Now we will see other variability. Okay, there are many genes that control specific traits and have more than two alleles. We have discussed that one gene has two alleles in its two homologous chromosomes, but there are some instances that some of the genes may contain more than two alleles. Okay, this means that there are far more possibilities for different phenotypes, and far more possibilities for different phenotypes means more variability. Okay, so let's check what is multiple allele. So in normal case, what we know that we have two homologous chromosomes, and in each chromosome for a particular gene there is a locus, and in the same locus there is another counterpart of the gene, another variation of the genes. So they are known as alleles. We know this already that for a particular gene there are 2 copies, and the alternative form of the 2 copies are known as alleles. So we from the previous experience we know that one gene contains 2 alleles. One gene, it's 2 variant, so there are 2 alleles, but there are some genes where more than 2 alleles are present in the population. Okay, so it is common for more than 2 alleles to control a trait in a population. Multiple alleles can only be studied in population since each individual has 2 alleles for a trait. For example, multiple alleles can be seen in human blood type, ABO blood type, where the 3 alleles are present: allele A, allele B, and allele O. But point to be noted, each human contains 2 alleles, but the different types of alleles are present in population. For example, what I can tell you, that if I am blood type A, I may have 2 alleles, capital A, or I may have capital A and O. Okay. If I am blood type B, I may have capital B, or I may have capital B and O. I nothing means IO. And if I have AB blood type, I may have 2 alleles, capital A and capital B. And if I am blood type O, I may have the alleles OO. Small I is also denoted as OO. Okay. So from here what you can see that there are different types of allele present, type A, type B, and here is type O also. So but in a single individual only 2 alleles will be present. Okay, but for the population level there can be many alleles, because one individual can have A, one individual can have B, one individual can have AB both, or one individual can have only O. So you can see here lots of alleles are present, A, B, O. So in one individual there will be 2 alleles, but in the population level there are more than 2 alleles. Okay, I hope you got this. So ABO blood type in humans is determined by 3 alleles, IA, IB, and small I. Small I is also known as O. Okay, you can also write small I or IO. Okay, with I nothing means that represents O, that O blood type. IA and IB are codominant alleles. We will come to this. And both IA and IB are dominant to the allele I, that is IO. There are other examples also, that is fruit fly I, coat color in rabbits. They are also examples of multiple alleles. So for example, I am telling you there are ABO blood type system. Okay, there is IA, IB, IO. Okay, these 3 types of alleles are there. IA and IB are codominant. I hope you understood what is codominant, that both the alleles will be expressed, and both IA and IB are dominant over IO. Okay, that means if IA and IO are present, the phenotype or the blood type will be capital A. If B and O are present, the blood type will be B. Okay, so here is also the case of dominance and recessiveness in alleles. Whenever there are alleles, there will be case of dominance and recessiveness. Okay, so there are 4 types of blood group we know: A blood type, A type, B type, O type, and AB type. It's very easy to predict O type. If you are O type, if you have O blood group, then your alleles must be IO, because O allele is recessive allele. You can see from here that IA and IB are dominant over IO. Okay, so if you are of O blood group, you must have 2 O alleles to have the O blood group. Okay, you must have 2 O alleles in order to express the O blood type, because IA and IB both are dominant over IO. So in order to express IO itself, it must have 2 copies of itself. Okay, and AB blood type, both the alleles will be IA, IB. We know that we know that A and B are codominant to each other. So AB blood type means IA and IB alleles are present. But in order to have a blood group type, you can have two type of allele combination. Either both alleles are IA, or one allele is IA and another is IO, because we know A is dominant over O, so the phenotype will be A blood group. And same the case is for B. It can be IB, or it can be IB, IO, because B is dominant over O. So by this we can have the blood group type. So the main thing is that we have multiple alleles, that are 3 alleles for human blood group type, but we in each individual have 2 alleles, but in population there are multiple alleles. Okay, so there are 4 type of blood type: AB, ABO we have already discussed. So from here you can read that AB are codominant, I have already discussed, and type A means type A, type BB means type B, AB means type A, ARB both are dominant over O. So AO means type A, BO means type B, because A is dominant over O, B is dominant over O, so that means AO means A, BO means B, OO and OO means O. So in order to press the O phenotype or O blood type, you need to have 2 O alleles. So A, B, O are the alleles. So there are 3 alleles for human blood type, so that is why it is known as multiple allele. And if A or B both are codominant, then when they are both present, they will be represented as A and B, giving us blood type AB. So this is one example of codominance, because A and B both are dominant. If both are present, the blood type will be AB. When A and O and B and O are present, you get AO and BO, but because A and B are dominant over O, you will get A type and B type. I have already discussed with you that A is dominant over O and B is also dominant over O. Okay, so I hope students you have understood what is incomplete dominance, what is codominance, and what is multiple alleles, a type of allele. If you have any queries, you can post it below in the comment section and I'll be happy to give you a suitable reply. You have to learn them by heart. Okay, rewatch the videos and try to grasp the concept, and if you have any problem, you can always revert back to me. Okay, thank you very much, students. All the best for your future. Thank you.